So I'm going to tell you about an application of Phage Display that I think is scaffold agnostic and which I think can make an impact on the field of diagnostics. So this is work that I've done at University of Illinois Chicago, and being an entrepreneurial effort that I'm trying to commercialize directly through a company I founded last year called Tango Biosciences, and the name comes from it takes two to tango. So as you all know, many assays, clinical assays, diagnostic assays are antibody-based, and it's a very large market, nearly $20 billion globally. And these tests are used for a variety of analytes, biomarkers that one can find in the blood, such as cytokines, human chorionic gonadotropin, a biomarker for pregnancy, insulin, biomarkers for a variety of cancer. And all of these assays have kind of a similar structure. They're based on having a pair of antibodies, I've labeled here antibody 1 and antibody 2, that create a sandwich with the target or analyte. So typically antibody 1 is immobilized in the bottom of a microtiter dish. One would wash, add the analyte, wash, add the secondary reagent that then has a reporter typically conjugated to horseradish peroxidase. And this assay format has a great deal of advantages. One is that it's compatible with many different biological samples, from cell lysates to cell CSF, blood, serum, urine. And one advantage is that the first antibody, which is immobilized, provides an enrichment function. So it can sit incubated with the sample and will then collect through affinity and specificity the target. And this format can be ported over to many different devices. We all use it for ELISA, certainly the dipstick for pregnancy tests, Luminex beads, proximity extension assay, or ligation, FRET. And one of the other advantages, which I want to highlight today, is in a way you have two levels of specificity. So while many antibodies will have some degree of cross-reactivity, I think that's just intrinsic to antibodies and the limited number of shapes or conformations that a protein can have or share. But the likelihood of the same cross-specificity overlapping is very small. And so that means you get very few false positives with these assays because the only molecule that will bind to both is the intended target. However, what led me to today's topic and presentation is realizing the disadvantages with this format. First, you have antibodies that are derived from animals, and every animal gives a different immune response. So once you've used up the rabbit antibody and go back to generate a second one, it may not have the same specificity or affinity. Second, as Andrew Bradbury and colleagues recently published, many of the hybridoma cell lines are actually secreting several different antibodies. And that secretion may change as the hybridomas grow, and so that ultimately there's a lack of full reproducibility and renewability with animal-based antibodies. And this is why everyone is switching over to recombinant approaches. Now, if you think about another limitation with respect to pairs, how do you find pairs? I would say that it takes time, money, and luck to find pairs. So for example, if you go to a catalog, a series of catalogs, and you look for all the commercial antibodies to your favorite protein, you would say buy 10, but you wouldn't really know whether those antibodies are the same or not. A dirty little secret in the field is that the same antibody can be sold by different vendors, and you can't tell from the specification sheets whether they're the same or different, or even what the nature of the immunogen was. Second, you can buy them, but you're really not sure whether they're going to be available the next year or the year after for the duration of your experiment. And so there is this possibility that what you find as a great assay will be discontinued, and then you have to go through this process all over again. So you buy 10 antibodies, you set up 45 combinations, and then you hope that one of them will work. Now, one other limitation is if they're both murine monoclonal antibodies, you need to be able to distinguish the second antibody from the first. So typically you would do some conjugation event. So you need to, if they're from the same animal or same isotype, you need to distinguish between the two. So at the end of the day, you hope you're lucky and you find a pair and then you move on. Now, how would this process differ by phage display? So here on the left, you have a library of bacteriophage M13, each displaying a different variant of a scaffold protein. You would do affinity selection for those that bind to the target. Here you see they're binding at 3 different epitopes. And then you would identify the binders from your output pool. You would characterize them, determine their DNA sequence, identify the unique clones, and then you would test them in pairwise combinations. But I would argue that even with phage display, you still come up to a barrier. How do you find out more than just a few pair? Because of the phenomenon of clonal focusing, with several rounds of affinity selection, you tend to have one clone that dominates. Another idea is that things that bind weakly tend to be lost at the expense of the dominating clone, and those might be the clones that you're most interested in because they bind at a different epitope. So what I've been thinking about over the last few years, in which I will present a solution, is how do you modify the phage display process so that it only gives pairs? What I've learned over time is that when phage display, you want to build into the process of affinity selection what you want at the end of the day, rather than to reconfigure your output for what you really want. So this process that I'll present, I call Megastar. It consists of phage display with some added elements that only gives pairs. So in Megastar, we do the traditional phage display, 2 rounds of affinity selection. We then reformat in a combinatorial manner the coding regions for the binders in that particular scaffold. And we create a secondary library that has tandem display. Then we repeat the process of affinity selection 1 or 2 more rounds with a reduced target concentration. And so essentially, we're selecting for variants that have a higher apparent affinity. And they have a higher apparent affinity because of avidity. That is that both of the displayed elements are contributing to binding to the same target. Or in other words, we find pairs. Okay, so let me give you some details and then some experimental proof. So the combinatorial formatting is a unique step to Megastar. We take our output clones and we do polymerase chain reaction across the coding region that differs from one clone to the next. This is the engineered variant scaffold. And then we take the double-stranded DNA, we denature it so that it's single-stranded. And that can be done by exonuclease treatment. One could make single-stranded DNA by asymmetric PCR. We just use heat. Then we anneal the single-stranded DNA to a vector that we've created that has 2 landing sites for the coding region. So the coding region is there present twice, upstream the truncated protein 3. And then this was in fact inspired by work by Greg Weiss, published in PNAS, where he noted that one could hybridize to single-stranded DNA more than a single oligo at a time. And so here we're annealing 2. And then by adding DNA polymerase, nucleotides, appropriate salt, you can convert this partial heteroduplex material into circular double-stranded DNA, closed with DNA ligase, and then electroporate into E. coli. It's very efficient. In a day's process, you can make a fairly large secondary library. And then the output secreted by the cells would then have the tandem displayed elements. One other point to talk about the vector. So the vector is a phage mid, 3+3 design, so it's a monovalent display, 2 landing sites for the green and purple coding regions. We've introduced amber and ochre stop codons into the 3 loops that we randomize. And so then, because when we generate the DNA, the only molecules that will have display the flag tag fused to the capsid protein P3 are where there's an open coding frame. And so that means we've got recombination in favor of the synthetic strand versus the parental strand. The parental strand is disfavored because it's uracil-lated. But the key point is that instead of testing 45 pairs, we're testing 10 to the 7th pairs. I also mentioned there's a linker. It's 25 amino acids long, a glycine serine rich linker. So one could have binding between E1 and E3 or E1 and E2, but to get binding between E3 and E2, you would need a longer linker. So I think that in discovering epitopes, one would want to have a suite of different size linkers to maximize their discovery. So I call this process megastar for mega primer because our primers are not 40 nucleotides long, but 300 nucleotides long. So that's the entire coding region of the scaffold. Shuffled, tandem, affinity reagents. The scaffold that we're using is the monobody. This is a segment of human fibronectin. We've randomized 3 loops. So the BC, DE, and FG loops. And this is a scaffold that's been popularized by Akiko and Shohei Koide. And so randomizing these loops is much like the 3 loops of a single chain of a VH or VL domain. And why I like to use it is that it's small, lacks cysteines, expresses very well in E. coli, is thermostable, highly soluble, and then because it folds well, it can bind to its target if expressed inside cells. So all the work that I'm describing is applied to this particular scaffold, but I think megastar is scaffold agnostic. Okay, so let me give you 3 examples. So this is a 30 kilodalton protein. It's a subunit of a ubiquitination signaling pathway. We isolated 60 different confirmed binders. DNA sequencing allowed us to group them into 5 groups. And so green would represent a family of monobodies that had conserved residues in the 3 loops, but with some variation. And I would then hypothesize that that represents a family of monobodies that binds the green epitope on the surface of the analyte. Green was paired with yellow 43 times. Yellow is another family of monobodies that we interpret to bind to a different epitope. So here we found 5 different pairs, and they would bind to 4 different motifs or epitopes. This is the P21 associated kinase, so PAC1. It plays a role in proliferation, cell migration. We isolated 49 confirmed binders, 8 different pairs, 7 different motifs. So they would recognize 7 different epitopes. Our best example was a Rho GTPase activating protein, 32. We isolate 15 different pairs, 10 different epitopes. So megastar is an epitope discovery machine. It identifies many different epitopes. And the idea is that these give many different pairs so that you can find the pair that works best for the experiment and assay that you're developing. Or in other words, more shots on goal. So let me show you that we do get avidity and that we do create pairs that work in sandwich assays. So here's a pair, green and red as blue, that was a tandem pair, isolating at 2 different epitopes. If we look at the binding isotherm of the variant that displays the 2 tandem pair as the individual, you can see that the tandem pair is shifted to the left compared to either of the clone alone. And the affinity is 21 nanomolar. Orientation does not matter. Here's a case where we found 2 variants, one that had green followed by red, or red followed by green. They bind equally well. And then the last is kind of a transitive property. If you find yellow paired with red and blue paired with yellow, would blue and red work? And the answer is yes. So this, not only the actual pairs that you identify, but you can then test other pairs that you didn't identify experimentally. And perhaps with deep sequencing, you would identify some variants that have that other pair. Okay, so probably the most convincing proof that these actually form pairs is that you can form a sandwich. So find a clone with the tandem monobodies, express each separately, and then show that you can create a heterogeneous assay. So a convenient method for us is to express one of the capture monobodies with an AVI tag, so that when it's endogenously biotinylated in E. coli, with BUR-A, it can be captured on immobilized neutroavidin. Then to add the analyte, and then we make fusions, genetic fusions of the second reagent, here in this case to nanoluciferase. And so one gets a signal that is analyte dependent. We've done the same as well using alkaline phosphatase as the readout. And one of the things that we've been able to streamline is that we're using reagents, the blue and red, without purification. So they're just in lysed E. coli. So the key differentiators are that we can identify more than 3 pairs for any given target. We're 6 for 6, so we have a 100% success rate. That epitope recognition is not limited by immunodominance or tolerance, 2 events that would occur in an animal-based system. One can do epitope subtraction, as Greg mentioned, a very efficient process so that you can distinguish between 2 family members by off-rate selection and competition. And that because they're sequenced and renewable in E. coli, one can also apply the latest and greatest tricks for protein engineering. So I'd like to acknowledge the members of my research team who are photographed here, and 2 prior lab members for their contributions to the effort, and certainly as well the NIH for funding various aspects of this project. And I thank you for your attention.